Good afternoon, everyone. As I was just introduced, I'm Rachel Nepia, and I'm here to share with you my research. And I hope that you will find it as fascinating as I do. Green button. So, with the major growth that we've seen in New Zealand's apiculture industry, the number of hives registered in New Zealand has doubled in the last 10 years, and on conservation land, that trend has exceeded the national trend, with hives increasing by 70% in the last 5 years. So with that in mind, it really is time for us to understand how honeybees function in native forest with regard to their interactions with native plants and other native flower visitors. My research over the last couple of years has been based in the KAMAHI range and is focused around 3 key areas. The first is understanding the interactions of honeybees with native plants, and that's massive, so I've focused in on just 2 species, Tawari and KAMAHI, which are 2 honey producers. The second part is how do honeybees interact with native flower visitors, and the third is understanding the availability of resources that are used for honeybee forage. So for my first research aim, part of that was using video surveillance to look at the floral parts that are contacted when flower visitors come to a Tawari or KAMAHI flower. The parts that will be most important for pollination are the anther and stigma, which are shown in that picture. So the graph describes the visits to Tawari at the top and the visits to KAMAHI at the bottom. The red bars represent the percentage of visits where only the nectary of the flower was contacted. The yellow bars represent where either the anther or stigma were contacted, and the green represents where both anther and stigma were contacted. So from this you can see that it's unlikely that honeybees would be effective pollinators of Tawari. They're quite large flowers, and the pollen-bearing anthers are at the end of those long filaments, so it's just too tricky. KAMAHI, on the other hand, has very small flowers, which are likely to be effectively pollinated by a range of different visitors. I conducted some pollination experiments at sites of varying hive density in the KAMAHI range, to understand the extent to which seed set of Tawari and KAMAHI was limited by the availability of pollen, and that's known as Pollen Limitation Index, or PLI. The honeybee icons on that graph represent where honeybees were 1% of the total visitors at that site. So at Tuahu, we had 10% of all flower visitors were honeybees, and that's where we observed the lowest limitation of seed set because of pollen availability. The other sites had less than 1% of visitors were honeybees and a lot higher pollen limitation. For the second research aim, over the last 2 seasons, I've collected about 1,600 flower visitors by sweep netting. The honeybees represented about 4% of those visitors. The most common ones that I collected were flies, followed by beetles, and then it was the bees and the wasps after that. I'm in the process at the moment of developing methods to identify the pollen carried by those flower visitors so that I can see which species are visiting which plants. I can then map those interactions in using network analysis to see which players in that network have the important roles and who's interacting with whom. This diagram just shows a simple cluster analysis from a very small subset of the samples which I've done so far. Different colours represent different groupings of the flower visitors based on the similarity of the pollen that they collected. So, for example, beetles, flies and bees like the same kinds of pollen, which gives us an indication that perhaps competitive effects could occur when resources are scarce. That leads to the third part, which is resource availability. So over the last 2 seasons, I've collected about 200 nectar samples from KAMAHI and Tawari, and I've observed so far that the average sugar produced per flower during the hot, dry 2017 summer was less than half of that during the cooler, wet 2016 summer, and that was a trend that I observed across all of my study sites. In addition, I conducted monthly counts of flowers and buds over the last 2 years at a range of sites and for a range of species. And this showed that flower numbers and bud numbers for trees followed kind of cyclical patterns between seasons. KAMAHI and Tāweri both had higher flower production per tree during 2016 than they did in the summer of 2017. And by the looks of the Tāweri buds that are out now, it looks like 2018 will be a pretty good year. So it's likely that Tāweri flowers follow about a 3-year cycle between the high flower production and the low flower production. This kind of information is really important because if you hit one of these low flowering years and a low sugar production year at the same time, resource availability could be a real issue. So in summary, this project represents just a small amount of progress towards understanding pollination systems in native forests, the resources that support those systems, and how honeybees fit into that picture. I think that in order to make this functional, it will need to be expanded into other forest types and into broader regions across New Zealand. But when we understand pollination systems and the way that honeybees interact with native flora and fauna, we can make sustainability achievable, not just for native forest management, but for native honey enterprises also. Thank you very much for your attention.